Okay, so this is actually a joint work that we did with Jean. I think we started maybe 2 years ago. Now it's almost finished, I would say, but still in progress for the application. So it's about, say, computational method to simulate system of cell populations in biology using optimal transport. So I wasn't completely sure about the audience and the background of everyone, so I tried to do a presentation that could be interesting for everyone. So just as an introduction of what I do usually, broadly speaking, I work on particle system and the question of emergence of large-scale phenomena. So initially I come from more the kinetic theory part, so it's the scale of the atoms, and here the particles, they are literally just atoms or molecules. And okay, so the interactions are maybe electromagnetic interactions, something like this. You don't care so much in the end, it's just physical laws, so conservation of energy momentum. And that's enough to have complex system of particles that contains basically all thermodynamics and fluid mechanics. So here you really have emergence of something large like fluid dynamics phenomena from just binary interactions between molecules. And then maybe over the last 2 or 3 decades, people have tried to apply the same principle to bigger systems, so at the scale of the matter, so for us, so to describe things like flock of birds or the school of fish, so all these collective phenomena that you can see in the animal kingdom. And the main difference between kinetic theory and social science is that kinetic theory, the laws are the physical laws, so you want to conserve energy momentum. For the birds, you don't want to conserve the energy because they produce their own energy. So if they want to move in a certain direction, they can do that. And for the particles, they are bound to, okay, the physical law. So the mathematics are much different. The phenomena are more nonlinear somehow, but the techniques are more or less the same. And what I want to discuss today is somehow the scale, a bit in between, which is the scale of the micrometer. So that's the cellular scale. So the scale, the typical size of a cell in our body. And there are many applications, in particular for development, so the embryonic development. So the simple question is, okay, we have the body is made of a bunch of cells, different cells. They have different behaviors. They want to do different things, but somehow collectively they manage to self-organize to form organs that have certain function. And okay, it seems that it works pretty well. So how do they do that? Because in a cell, basically they can do 2 things. They can either migrate somewhere or they can divide. And just with migration and division, somehow we have all different behavior, biological behavior. But even describing the motion of cells is not so easy because okay, for the molecules, they move just by conservation of energy. The birds, they move because they want to fly somewhere, but the cells, they don't decide to go somewhere. So it depends a bit on the mechanics. So if you want to see a cell moving, it moves a bit like this. So that's one cell. And you see that it makes some protrusion here and there to explore the space. And that depends a bit on the substrate, where you have to grab something to move. So they have to change their shape. So it's a kind of complex process, internal process of the cell to change their shape and be able to move somewhere. And that's pretty efficient. So if you attend talks in microbiology, the first slide is always this very funny video where you see like it's a white blood cell chasing the bacteria. Okay, that's very funny. At the end, the bacteria will be caught by the cell. But you see that this cell migration is quite complicated, where you have to move across obstacles. There are red blood cells, and you have to change the shape to somehow be able to go to the interesting place. So that's just for cell motion, is already something difficult to describe. And then there is division, so it's mostly for during embryonic development. So this is an embryo of C. elegans, which is a kind of a transparent worm, so it's very easy. You can just see in the microscope. So there is 1 cell at the beginning, and then 2, and they divide again and again. And if there are 2 cells, it's easy, but then if there are 3, 4, then they have this special arrangement, right? So it's not just division, you have also to fit the space that you have. So that's also another complex thing that we would like to model. And eventually we want to understand why the tissue look like what we have. So this is a typical image of a tissue. So I think that's the, if I remember well, the eye of some fly, where you see all the cells and they have this kind of more or less hexagonal shape, I don't know. But there are tissue with different shapes. So that comes from textbook, basic textbook in biology, where you see that the tissue they can be made of very long cells or very small cells, and depending on the shape of the cells, the tissue will have different functions and also different physical properties in terms of mechanics. So that's a bit the typical system that we would like to model. So multicellular system. So let's say just an aggregate of cells of different type, but in 3D, because development is typically in 3D. So the examples are shown somewhere in 2D or 3D, depending. So you have all the cells, they have their own morphology, they have their own dynamics. So they can be big, small, have different aspect ratio, they have different properties. They can be very hard cells or very soft cells. And then the interactions, it's mostly mechanics and a bit of chemistry, because it's not like birds, right? There is no social science here. There is a bit more than just physical law, so it's a bit in between. But with just that, we want to understand self-organization. So how do we have sorting and spatial arrangement? So this is another video of an embryo. So you will see like two types of cells, green and magenta, and they are all mixed together initially. And then with the development, well, they sort, and you can see at the very end, the spatial organization is not so trivial, where you have this hole in the middle that's actually what we are, right, basically. And this spatial organization comes very early in the development. So of course, if you want to make experiments and if you want to have this kind of beautiful 3D imaging of the embryo, it's quite costly and quite complicated when you have to make embryos, and most of the time it fails, so you have to start again. So it would be nice to have good computational models where you can just simulate, because it seems like something that not crazy to simulate, right? There may be 1,000 cells. Okay, it's in 3D, but mathematicians have done things more complicated than that. So can we find like good computational model that would save a lot of time and money, and then we can explore the role of each parameter to see what is critical to have that kind of self-organized behavior. So what I will present is somehow yet another computational model for that. I will briefly mention some important models that are still very useful. I mean, it's just a review, but also to show for what kind of problem, what kind of method you can use, if it works. So historically, I think in biophysics and computational biology, people have started to work with cellular automata. So you just take the space here in 2D, but can be in 3D. You discretize and you just say that, okay, if I have here in that example, 4 types of cell, 1, 2, 3, and 4, then for each cell grid, you say that it belongs to cell number 1 and cell number 2. And then you want to just switch and say, okay, maybe this cell will expand that direction or in the other direction. So for that, you define some Hamiltonian, so just an energy. So the typical energy is that for all neighboring cell grid here, you define some interaction potential J that can model anything you want. Can be just a cell-cell adhesion typically. And then you have other constraints. So here it's a volume constraint. So the little v would be the volume of the cell type sigma I. Sigma here in this example is 1, 2, 3, and 4. And v is the target volume that you want. So you just add that, you create that big energy. And then based on that energy, you can just flip each cell of your grid to one or the other state. So it's a somehow classical metropolis procedure. Yes?
So is it?
Yes, yes. So sorry, so sigma would be 1, 2, 3, 4, and tau would be the type. So here, for instance, there is this yellow and red cell. So you have the ID of the cell and the type of the cell. But it's here it's written very abstractly. You can put just to show that the typical shape of a Hamiltonian would be like this, but then you can put basically whatever you want. So it works pretty well, but okay, in terms of physics or biology, it's a bit arbitrary, right? There is no physical law per se here. It's just very computational. It's very efficient and easy to code, but okay, can we do something different? So other methods, and somehow it's the simplest method of all, but that's maybe my favorite somehow. It's just molecular dynamics. So the cell, okay, they have complex, they are complex entities, but you can just say that they have a position, they have some size, and then size means that basically attraction radius or repulsion radius. And then it's just a molecular dynamic simulation. You just write a Newton equation for the position, and you don't care at all about the shape. You are back to very basic kinetic theory simulation or molecular dynamic simulation, where you don't care at all about the shape or the volume. You just hope that if you find a good interaction energy or interaction forces between these point particles, then you can obtain something interesting. So here's an example where you have each cell would be just one ball, and you can grow different types of shapes. So that kind of model is actually very useful for morphogenesis. And I thought I would talk about that, but then you changed my title.
Sorry,
No.
No.
No. It's actually the project is not completely finished, so it's okay. It's better to talk about that. It's just to say that that kind of very basic model where you just have a position and some interaction, and you believe that you are just doing elementary particle physics, then you can still apply that to morphogenesis, but you have to be very careful of the kind of how to encode the mechanical and chemical interaction in a good energy. And computationally speaking, okay, the drawback is that it's a bit costly, because if you want to simulate an organ, then you have thousands of cells, and you have to have 1,000 square interaction to compute to simulate the full organ. So of course, Jean here has developed this beautiful library Cheops that can do that very efficiently. There are people from NVIDIA that have developed this position-based dynamics that works pretty well also. So there are many improvements to cope with the problem of, okay, it costs a lot to implement. Okay, so then maybe, I think one of the most popular model, even in the literature when I checked, is this vertex model or the Voronoi model, that are a bit different, but philosophically similar, I would say. So you want to move to something a bit more realistic than just a point. So you want to have a shape, so an area and perimeter. So it's basically just a partition of your space. So you can define each cell by just the set of vertices, and then and define the target area and target perimeter that you want. Again, you define an Hamiltonian, so that would be a neutral one would be that you want the area of the cell I to be close to this A0 and the perimeter to that, and then you have two parameters that you can choose freely. And if you minimize that with typically just a gradient descent on the parameter, so the parameter would be the position of the vertices, and in the Voronoi diagram would be like the center of the Voronoi tessellation, then it gives something, and something that is quite realistic. So here's the simulation of just cell growing, and I think the color is the pressure, and here's just random motion in some kind of not completely saturated space. But that's mostly 2D, right? If you want to do that in 3D, then it's a bit more complicated. So in 3D, people work more with finite elements, which essentially is the same as a vertex model, but the vertex is not the vertex model in 3D would be a 3D cell. So here you work with the mesh, but then the mesh, you also define the mesh in 2D by a set of vertices, some well-chosen Hamiltonian that you minimize with a gradient descent. And so that works pretty well in 3D. So it's a simulation of just a cell dividing and growing. So that makes this huge ball with thousands of cells. And I think nowadays there are very fast implementations for that kind of method. So that's a bit of summary of the existing method. So the simplest one are the point particle system. Then you have cellular automata, vertex model, finite element. I haven't talked about different methods that are more phase fields. So I think they are not so much used. They are somehow efficient, but computationally very demanding. And theoretically, I think even more. Okay, so I was a bit at this stage when I started this project with the biologist I was working with. We had like this imaging data with cells. It was dynamics, and okay, can we find a model where we have a good description of the cells? They can change the aspect ratio. It can be dynamics, etc. Can we fit with the imaging data? So since the beginning of my PhD, when I don't know how to implement something, I ask Jean and ask him, do you have idea? And he said, oh, but that actually the data that you have, it looks very much like this steel industry problem where these are not cells, they are just crystals. I think they call it like this. And the crystal, they form not a tissue, but they form just steel here. And depending on the shape and the volume of the different crystal, then the material can have different mechanical properties. So it's of course very important for industry. And so I think Jean is working with David Bone, who developed this optimal transport model for steel industry. And the idea was actually to borrow that idea and apply to biology. But here the context is a bit different because they want something very static. So they have image like data of steel. You can correct me if I'm wrong. They have data of steel, and that's a fixed image, and then you just want to fit a model to that image. So in biology, it's a dynamical process, right? The cell, they move and they grow and they divide. So you don't want to fit just one data. You have to just one image. You have to have a dynamical model. So okay, so I started exploring this optimal transport model. So that's a bit what I will present today. So the main assumption that I will make here, and that's almost the only assumption, is that I want to model the cells. So I will call them particles for, I don't know, I just used to call them particles. Particles with just their volume. The only thing that I want to treat as a hard parameter is the volume, because that's basically what you have in the data. If you have an image of a tissue, the first thing that you can measure is the volume. So that should be somehow the input of the model. And then I will describe a bit this optimal transport approach for describing tissue, and then how to control a bit the shape and deformation of each cell, and how to turn that into an active matter model. That is useful for biophysics, so we can implement some biophysical laws that would lead to all the self-organized phenomena that I discussed. And at the very last, because it's my field originally, I would like also to discuss more, very briefly, some more mathematical idea of are there links? Is it only a computational model, or can we actually do some mathematical analysis, in particular for partial differential equation analysis? So what the model that we want to construct, it has to be a compromise, because you cannot be physically accurate, you cannot have the fastest computational speed, some kind of biological versatility where you can model different phenomena and at the same time do some mathematical analysis. So you have to simplify something. It's a compromise, I think, between these 4 points. Okay, so I guess there are many experts in optimal transport in the room. probably more than me, but I will still just explain briefly how to link optimal transport with this volume constraint problem. So let's say that I have a particle at a given position in dimension 1. So the space is this line here. And I want this particle to have a given volume. So of course in dimension 1 is a bit trivial. It's just a segment, and the segment has some length, and okay, that's the length is the volume in dimension 1. But what we can do also is to say, okay, we take a cost function, so a function that is not too flat. And you see that if you check all the level sets of that function, there will be some value f, where if you want the volume to be 2.83 here, then there will be a level set that corresponds to that volume. A level set meaning that every point below that threshold belongs to that particle. So instead of measuring the length, I can just measure a level set of that function in dimension 1. So for one particle, it's of course, okay, it's easy. As long as you have a cost function that is not flat, then you can see easily that there will be a unique solution. If you have more than one cell, it's not so easy because, okay, here I have two. I want to tune their volume. So I do the same thing. So here I just fix the level set and I lower the potential. But you see at some point, if they want to expand, they will collide. So if I increase the volume of that red cell, that will decrease the volume of that green cell. So can we always find two value f1 and f2 so that we have the right volume constraint? It's not so clear. It's even less clear if you have more than two cells and if the costs that you choose are different. So hopefully there is a theorem that says that there is a solution and it's unique. So it says that if you take positions and volume, so omega is some compact set, the volume is just a non-negative number. Then if you take a function c, which is the cost I told you, that is not too flat, so you can think of just typically the L2 cost, so the square function, then you can always find a set of n potentials, so real numbers, so that you can form a partition of your space that satisfies the volume constraint. And on top of that, there is an expression for the partition. So the space is partitioned into this subset that are called Laguerre cells here. And it's a set of points that satisfy a set of inequalities that involve, of course, the cost and the potentials that are constructed by the theorem. So if you look carefully at the definition of these Laguerre cells, if all the potential are 0 and c is just the distance, the Euclidean distance or the square Euclidean distance, then that's the definition of a Voronoi diagram. So to some extent is a generalization of Voronoi diagram. And probably one of the first person to notice that kind of property is Jan Brenier, but it was for fluid mechanics. And 1 or 2 years ago, I think Bruno Levy came to talk about that in that precise seminar to say that, okay, by doing that, we can actually do fluid simulation. And the way that fluid simulation works is that all these particles are just fluid particles. So basically, you construct a mesh that satisfies volume constraints. So it's very good, of course, to simulate incompressible flows. And I think Bruno Levy showed that beautiful simulation, fluid simulation of just a droplet falling in water, and that it's a mesh, and the mesh is made of this Laguerre tessellation. And here, when I realized, oh, okay, initially Jean told me about these steel industry applications, and then I realized, oh, okay, so it comes actually also from fluid dynamics, and biology is exactly in between. We don't want to construct a super fine mesh like fluid mechanics, but we want something dynamical, not as steel industry. So basically, we want to have like a big mesh, but dynamical in space. And the link with optimal transport, because so far I haven't talked so much about optimal transport, is that, okay, finding this potential F that are called W, unfortunately, on that pictures, but That's the same as before. Then here you want to construct like a partition of the space. So you can recast that as a minimization problem over the set of partitions. But okay, that's probably difficult to minimize over a set of partitions. But actually, this problem is equivalent to an optimal transport problem, where and the optimal transport is between the Lebesgue measure, so the ambient measure of the space, and a discrete measure, which is this sum of Dirac masses at the position that you want, and weighted with the volume. And if you solve that Monge problem, so if you find a map that sends the Lebesgue measure to the discrete measure, then you can see that you construct a partition of the space, because each point of the space here in blue will be sent to some point here in red, which is the discrete point where you want yourself to be. So by doing so, then you naturally construct a partition. And the magic is somehow that the partition that you construct is exactly the solution of the minimization problem over the set of partitions. And it's very good because then many people in this room know how to solve optimal transport problem on a computer very fast. So here what we used is actually we had a discrete, semi-discrete optimal transport. So you want to transport a continuous measure, the Lebesgue measure, to a discrete measure, this set of points weighted by the volume. But the Lebesgue measure is a bit complicated because it's continuous measure, but you can always discretize your space. And then if you want to solve the optimal transport, then if you discretize the space, then the Lebesgue measure is the sum of Dirac masses, and you want to just match the Dirac masses together. So it becomes a fully discrete optimal transport model. And you can apply many well-known methods. So I'm very quickly on. I explained that very quickly, but the algorithm were already written essentially.
So maybe from an outside perspective, I think what you describe at first is that you want to work with cells, and the big question is how do you define easily a data structure where you are tessellating the space between like the ambient medium and the different cells, and the cells should never intersect each other. And that's something which can be hard if each cell is encoded as a discrete surface mesh, right? You have to test for collisions all the time. Basically, it's complicated. And I think
that's a complicated part of finite element.
Exactly. And so then the question is, what kind of data structure could you use? One idea would be to use Voronoi cells. Because Voronoi cells, in computer graphics, you have faster algorithms to compute them, etc. So you naturally tessellate your space. The problem is that with Voronoi cells, you have no way of prescribing their volumes, which is problematic in biology. And there, the only thing basically to understand is that the theory of optimal transport, you can interpret it exactly as giving you a fast algorithm to offset your Voronoi cells so that they have the volumes that you want. And then you close the optimal transport textbook and you use that to do whatever you want with it. But it's really this data structure of semi-discrete optimal transport. It's Voronoi cells plus volume constraints. So
yes, and the optimal transport part here is solved as a discrete problem, actually. Also another improvement that you can have, because otherwise it's not so clear how to implement that.
Sorry, in the end, is it a hard constraint or is it just a...
No, it's a hard constraint. And that's maybe also the main difference with what I discussed before, the vertex model or the cellular automata, where you can put the volume as a constraint, but that's a soft constraint. You just put that in your Hamiltonian, and if you want the hard constraint, then you have to, there is a Lagrange multiplier and you want to send it to infinity, but that's always a soft constraint. Here it's a hard constraint. So the partition of the space that you construct always has the right volume.
And it always exists.
Yes, and it's always unique. That's otherwise that's less interesting.
Under very mild assumptions, like the sum of the Prutride volumes should be smaller than the volume of the ambient space. So you just check that.
But here the x's are fixed, they are known.
Yes, they are known, but they are known at a given time. So at a given time, if you look at your tissue, they have some volume, but then you can say that the volume will evolve in time. If you want your cell to grow, then you just add a dynamic on the volume, and each time the volume grows, then you compute a new tessellation anyway.
So you have a system that evolves.
Yes.
The
So that's the somehow.
at all times.
Yes, exactly. So here it's a static version. At a given time, I have given volume. Doesn't mean that over time the volume doesn't change.
And if you want to add cells, is it going to explode or
Sorry, if you
want to add cells?
that's the same. At a given point, you have a certain number of cells at certain positions with certain volume, but then you can, at the next time step, you can add a new cell anywhere with another volume. You will recompute the partition. No, because the theorem tells you that you can put the cells anywhere. They can have the volume they want, as long as it's not too big, right? There will be a unique partition. So in it yes.
Sorry, but you cannot track one cell then. How do you track the cell? Because you have a bunch of points, another bunch of points.
But the cell is the you transport the Lebesgue measure, so that's a set of points you don't care, to the position of the cell. So that these points you always have them.
Yeah, sure.
So these points you don't care, it's just a numerical method. Okay, so the model summary somehow, okay. So at a given time, you have n particles given by their position, so just point in space, with their volume. The volume should be smaller than the volume of the space, of course, but it can be strictly smaller, so which means that around the cell there will be some void space. So that's a bit annoying, but you can always say that the void is another particle, and the volume of that additional particle is the remaining volume of the space. So here the space is the total volume of the space will be 1, and then if the sum of the volume is smaller than 1, then you add another particle that has the right volume, and you call it the void. And then for each particle, you have to choose a cost function, which is the function you use the level set of which you have to find to construct your Laguerre tessellation. So that's an arbitrary function, but just for the void function, you always take 0, which means in terms of transport that if you want to move a point of space to the void anywhere, it doesn't cost anything. Okay, and then each particle is represented by this Laguerre cell. So the next step is to understand, okay, the cost now is a bit arbitrary, so if we change the cost, what does it change, right? If I take the L2 cost, it's natural, but what about other costs? And then the question of how to turn that into a dynamical system. So the basic loop is that I compute a Laguerre tessellation. Then given that tessellation, I can compute interaction forces, or that depends on the tessellation that are just given. So that's just a computation. I apply the forces on the position, exactly like a molecular dynamic simulation. So I move a bit the position. Maybe I also update the volume. I add new particles. And with this new data, I compute a new Laguerre tessellation, and then again and again. Okay, and the question is, okay, can we find good costs and good forces so that we can actually simulate biophysical systems? So for the first part, so how to choose the cost. So the nice thing with this model is that we know that the cells are given, or the particles are given by Laguerre cells, and we have some algebraic equation for that. So we know that the intersection between two Laguerre cells, so the boundary between two Laguerre cells, would be given by this algebraic equation. And then if I want that to be a straight line, then okay, I know if the cost is the L2 cost, is the square distance, that's the equation of a straight line. If I want the intersection, so the boundary between two Laguerre cells to be a circle arc, then I take just the distance, the Euclidean distance, and that's the equation of a circle, basically. And then for one individual particle, so in the void. So the void has cost 0. So this equation reduced to that kind of level set equation. So basically the boundary of a particle in the void that define the shape is just the level set of some function. But then if I want my particle to have any shape I want, I mean, not any shape, but a star shape at least. If I want the particle to have the shape of a star, then it's very easy to construct a function such that all the level sets are scaled version of that shape, right? And what the optimum transport part will do is find the exact level set that has the right volume. So by changing the cost function, I can also change the shape of the cell. So that's a bit what we did to simulate like tissue, like with different cost function. So this is a L2 cost function. So you see that the interface between two Laguerre cells are straight lines. So it looks like a Voronoi diagram, but with volume constraint. Here it's a more bubbly diagram. So you take just the distance and then they look a bit more like disks. And you can even put something random, right? And then the boundary will look very random. So here the shape depends on some ambient field. So here they are more elongated towards the center of the field. Here more, it's the opposite, orthogonally to that field. And here we'll discuss a bit later. Here it's in 3D where I set the cells to be not just balls or have not symmetric shapes. They have some aspect ratio and I want to fit them into a big box. So by just tuning the cost, I can somehow control the shape of the cells, the interface between two cells. Okay, and then the dynamical framework is, okay, how do I evolve the position of the cell? So before adding any biophysical law, so biophysical law would be like surface tension between two cells typically. The natural idea is that, okay, in the slide before I said, oh, this is actually a minimization problem for some cost, right? And this cost, I can also take the gradient descent of that maximal cost. And what does it do? So it has a nice expression actually. The gradient of that cost is the integral over the Laguerre cell of the gradient of the cost of the particle. And that if you apply Stokes' formula to that equation, then the integral over the Laguerre cell becomes an integral over the boundary of the Laguerre cell. And here you can see that expression as a kind of internal pressure law. So what happens is that if you have a Laguerre cell that has this shape, so around is just a void, here is the centroid, so the xi. For each point on the boundary, there is a normal, right? And on the normal, then you weight that point by the cost that you choose. And it's a bit like you integrate all these elementary forces that are like internal pressure force to compute the total force, and you apply this force to the centroid. So it's a bit like kind of internal skeleton that will move the position of the particle so that the particle will restore its preferred shape. So this is a simulation where I have 4 cells in the void. It's the L2 cost. So the L2 cost, I said before, so if you take the L2 cost here, the shape of the L2 cost will be like the square distance is constant, so the basic shape will be just a disk, and the intersection are straight lines. So that's what you see here. And if you compute the gradient of the total cost, and for each point, so the gradient with respect to the position of each Laguerre cell here, then the force balance is like this. And if you run the simulation, they will just repel each other. So this is a kind of a repulsion force. You can see it as the internal pressure of each Laguerre cell will just create a repulsion force. Yes.
But you do need to have a very specific cost function to be able to get the actual boundary and get the integration, or is there a way to?
What?
If you want to compute this integral, you need to define like the surface.
But
is it because
basically we don't. You never compute that because you don't want to integrate over the boundary. What you compute is this, because then the gradient of the cost easy, and then you integrate over the volume. But the interpretation
Yeah.
Integration of a volume, not so clear, but physically integration of the boundary is more so that. But yeah, in the computer, it's only this expression. Okay, and then so I have this basic internal pressure force, but I can add any other force I want, right? So if I want to have a self-propulsion, gravity, attraction between points, and later we'll discuss surface tension, blah, blah. Then I can all add that in that equation, always with this repulsion force, even some noise, and then have basically an active matter model. So this is yet another simulation where I have just balls and they are falling. So I have just a gravity force and they are in that weird environment. And I don't do anything else than computing repulsion forces with the force I just showed before, plus the gravity. And they somehow repel each other in a quite realistic way, I think, at least visually. Okay. And I can also do the experiment I showed before with finite element, where I have just different rules with growth. So here the volume will change in time. They divide. So basically, I start with just one Laguerre cell. I divide in two, so they have the volume is divided by two. Then the volume grow and they divide again. And if you do that, then you have exactly the same kind of dynamic that I showed before, where you have an aggregate of balls that grows until they fit all the space. And then that's the part where the computer explodes, so you stop. Okay, so that was to show that by just having this internal pressure force, I can add any other force. So now I want to discuss, okay, if I change a bit the cost, what does it change dynamically? So what I did is one of the simplest model in biophysics, which is active Brownian particles. So you just take particles, they move randomly in space, but they are active, so they move at a fixed speed. And there is repulsion between particles. So of course, if there is no repulsion, if they are just particles moving randomly and they don't interact, then you know it's just a diffusion. And then if you have interaction, then what does it happen if they repel each other? So I just take some costs which are the distance to some power alpha. And if alpha is very below 1, then the cost will be concave like this. And if alpha is much larger than 1, it will be more concave. And from the definition of the Laguerre cell, you see that, okay, if you want something very deformed compared to the basic shape, which should be a disk, then you have to pay a lot. But you have to pay a lot only if the cost is very convex, because if the cost is very concave, if you want to say a point that is very far, if you want to match a point that is very far to your Laguerre cell, then you won't pay that much more. So if you simulate this system with different costs, with different alpha here, with a very low alpha, then you have cells a bit moving randomly here. Okay. And then if you increase alpha, it becomes a bit more rigid, right? They cannot move so much because they repel each other. And if alpha is really big, they really, really like hard sphere. So they even reach that kind of hexagonal configuration where they cannot move because they repel each other too much. So by tuning the cost here, I tune the softness of the particle, which is one of the basic biophysical properties of the cells that we want to control. Okay, and then I say that by tuning the cost, I can change the shape. So in that experiment, somehow the cost imposed the shape, like the shape of an individual particle here would be just a disk. Okay, but then if I move away from just the Euclidean distance to some power, I can find a cost such that the level set of that cost will have any shape I want. So typical shape would be this kind of a spherocylinder, which is the shape of some bacteria. And then this has another, this shape has another parameter, which is the elongation direction, so the principal eigenvector. So if I run a bit exactly the same experiment here, without the noise, but instead I just change this parameter, which I always take to be the principal component, the principal eigenvector of the shape that you can compute, right? If you have a tessellation, you have shape, you can always compute the principal eigenvector of your shape. And then you let them run actively at a given speed. Then, sorry, what you get is that kind of system where you have this spherocylinder, they move in straight line, but sometimes they bounce at each other, and it's chosen so that when they bounce at each other, they turn a bit, and then in the end they align. So that's a kind of a more or less realistic simulation of swarm of bacteria, actually. And for that kind of cost, you can do the same thing as before, right? If you take that cost and convexify a bit by just raising it to some power, you can have very hard spherocylinder or very soft spherocylinders. Okay. And so
So in this example, each particle has its own cost.
Yes,
It's not the same, so you can have
they have a. It's the same function, but with different parameters. So the two spherocylinders is the same function, but I mean, you just rotate. So you just add one more parameter. So now it will be position, volume, and elongation direction. And that's enough to define the cost function.
But you could have two populations of
Yes,
cells.
yes. But analytically, it's the same function. Okay, and now in the remaining time, I will just describe a bit the problem of implementing surface tension effect, which is a big problem in cell biology, because it's believed to be the main driver for cell sorting. So I already showed you this experiment where you have two types of cell. It was for the cellular automata, you have like red and yellow cells. And depending if they like each other, so if the red cells like each other a lot, they will cluster and the yellow cell will be around. So I think that's what happened. And that happens all the time in cell biology. You have different populations, they have different affinity together, and just that creates some sorting. So that's called the differential interfacial tension hypothesis. That has been done in some computational model long time ago, actually, in the early 2000s. And these are actually real pictures from biological experiments where you have two types of cells, and you see if you start with something very mixed, then with just surface tension effect, they can cluster and create these very well sorted patterns. So here it's completely engulfment, here it's a bit more partially, and here is the opposite. So you imagine that the green cells here, they like a lot the black cells, and the black cells, they like the green cells, but the green cells together don't like each other. So that creates that kind of checkerboard pattern. So can we do that with that model? But basically, we have to choose a cost, and we have to choose forces between cells. So the cost here, I just take the L2 cost for simplicity, scaled with some kind of volume, but doesn't matter so much. And then the forces, I spent actually many weeks or months, I don't know, thinking what should be the right forces. So in the end, I think we came up with that idea that, okay, if we want to minimize the curvature of the interface and the interface area, that's basically the two main effects of surface tension, then you can define a force which looks like this. And basically here, so you have the curvature, so you integrate over the interface between two cells, and what you integrate, so the direction is the normal of the interface, and what you put inside is the curvature and the inverse of the distance. So this first time would say that if the curvature is high, the force will be higher, so there will be higher repulsion between the cells, and if the interface is too large, then it's the same. So that's very arbitrary somehow. But if you look at this expression, you can compute analytically. If you have only two cells, that is easy, right? If you have only two Laguerre cells, you can compute the equilibrium. And the equilibrium is exactly what the surface tension equilibrium would be. It's given by an equation called Yang-Dupré here. And so you have two cells with different colors here. There is some adhesion between them, but not so much, so they don't merge, right? But they stay a bit stuck together. And the way we measure how stuck they are together is this angle theta. And if you apply basic principle of physics and compute what should be the effect of surface tension, you know that this angle theta should be given by that equation where the eta and gamma are the surface tension parameter. And if you use that expression of the force, that I agree comes from nowhere somehow, then it's the same equilibrium. So I believe, okay, maybe we are not too far, so let's try to make simulation. And with this simulation, we want basically to reproduce this kind of patterns with engulfment, sorting, and partial engulfment. So sorting is easy, right? You just need to say that the orange particles, they want to be together, the blue together, and they don't like each other.
And so to be clear, you are choosing what is a gamma orange.
Yes, yes. Yeah, sorry, I should say.
Yeah.
Thank you. So to tune the surface tension parameter is this gamma and eta. And the gamma and eta would just define who likes who, right? Because you have different gamma between two types of cells. So here the addition or repulsion, depending on how you think, between orange cell is much higher than between orange and blue cells, and between blue and blue cells also much higher between orange and blue cells. So here you have complete sorting, but you can also have this checkerboard pattern where the blue likes the orange and the orange likes the blue, but the blue and orange don't like each other. So it's a bit more mixed. And you can have engulfment. So here you just say that the orange and orange, they like each other a lot. And the blue, they like the orange. So that just creates that engulfment.
Is it hard to tune?
That's the next slide. So it's not hard, it's just that you have many parameters. So fortunately, I have a big computer. I used to have a big computer, so you can just run a bit all the parameters and you get a bit all the patterns you want. So it's hard in the sense, it's a bit arbitrary, right? I cannot measure something experimentally and put that on the computer. Depends a bit. I don't know exactly what parameter I should put, but if you explore the space of all the parameters, you have all the patterns you want. Then you just pick the one. Yeah.
It's interesting because it's also a motivation for fast numerical models.
Yes,
Is that they allow you to compute those kind of phase diagrams,
exactly.
which you could not do if a single simulation was taking minutes to run.
Exactly. So here I run more than just that.
So
That took a few days.
The boundary, there's no like weird fractal effect of phase transition.
But there is a phase transition if you want. I mean, that's a phase diagram. But no, there is nothing very weird happening. If you check all the patterns you get, maybe that's a bit weird. I don't know. But in the end, it's always a mix between engulfment, checkerboard, and sorting at different level. Okay, and here I also answer your question you asked before. Here, to compute that force, it's an integral over the interface. So you have to compute the interface. And for that, you have to compute a mesh. But the mesh is only to compute this force. And then, again, I asked Jean, but I want to compute integral over interface, how to do that? It doesn't work because now we have a volume representation. So he say, no problem, just open the VTK documentation and there is the algorithm you're looking for. So that's how we do.
Yes, so it's actually super useful. You know, in medical imaging, it's very common that we have volumes which are segmented, but with many different labels, not just one. You know, and five years ago, maybe you would use marching cube and marching cubes for every single label, but then if you have 100 cells, 100 times marching cubes, it's not realistic to do it at every time step. And since it's so important for medical image visualization, the people who work at Kitware, they really worked on a very fast volume to mesh algorithm that is SurfaceNet 3D, which is implemented in VTK, accessible in PyVista. I think it's the contour labels functions, and that's really a game changer for those kind of back and forth.
And the cost of doing that is less than the cost of solving the optimal transform problem. So you don't see it in practice. Okay, so it's almost done. So that's another, it's just for fun. I just wanted to simulate bubbles. It's basically the same as before, but they have different volumes or different shapes. So I say it's just toy example because there is no physics here. It was just to see if we do that, does it look realistic? Okay. To conclude, I think just 2 minutes, because I still wanted to talk a bit about PD, is to say, okay, at this point, it's only a computational model. So it's interesting. We can do a lot of experiments. So, but do we have more can we make more analysis on that model? So the thing I'm familiar with is this, say, mean field equation or Makin-Vlasov equation in particular. So Makin-Vlasov equations are just equations for interacting diffusion processes. So diffusion process is given by a stochastic differential equation that links the derivative of the position with the Brownian motion, and then the deterministic drift part is whatever you want. But whatever you want, that depends on somehow the empirical measure. So with that kind of very abstract shape, you can actually model a lot of physical law, just gravitation fits that framework, electromagnetism, all these binary interactions usually fit that framework. And that we know that when we have a lot of particles, we don't want to track each individual particles, because if you are in a gas, typically, then there are like billions of particles. So you don't want to track each particle individually. You want to have a more statistical physics description of that. And the statistical physics description of that is given by a Fokker-Planck equation that you can derive from that huge system of stochastic differential equation. So when there is no interaction, it's just Ito formula, which is the first thing you see in stochastic analysis textbook. And if there are binary interactions, then you get other type of equation, in particular granular media equation, but here it doesn't matter so much. The question I had is, okay, can we do the same with the equation that we implement? Because in the end, what we implement is a kind of stochastic differential equation, except that we add this additional internal pressure term that model this repulsion. And that doesn't completely fit the usual framework. At first, that's what I thought. But then if with just formal computation, you can actually recast that into a more traditional Makin-Vlasov framework. And the equation you get by just doing the same procedure formally here, the theorem is not written. So this is a theorem. This is not a theorem. Then is a Fokker-Planck equation, but which is coupled with another equation, which is a Monge-Ampère equation. So for the expert in optimal transport in the room, probably it's not that surprising. Actually, there's a very similar system that was written by Jan Brenier a long time ago, but I haven't seen so much analysis for that system. And in the system that Jan Brenier wrote, the cost was always the L2 cost, because it was more for plasma physics applications. But here I said, we said that, okay, if we change the cost function, then we can have a different behavior. So in particular, I said that if we simulate like this active Brownian particle, we have more soft or fluid particle. And so if I take this same equation that fits perfectly well this framework, this alpha was the softness parameter. See, when it goes to 0, there are no interactions, so I expect that to behave like a pure diffusion. And when alpha goes to infinity, I expect that to behave like hard spheres. So how does it translate in the equation? The answer is I don't know. But if I do is the same, then I did another simulation which is almost the same, but it was kind of a simulation of the evacuation of a room. So there are all these people, they try to reach that point. And if alpha is 8, so they are very hard, then they are a bit blocked, right? And if alpha is small, they behave more like a fluid, so everyone can escape because they squeeze. And that's very close to a system that has been introduced by Bertrand Maury, I think. And they have a nice PD description for that. So they did a simulation at the particle level and said that the good equation should be that continuity equation with a congestion constraint. So it doesn't matter so much what's the form of the equation. The question that I have, and then I asked a colleague and he told me, yes, of course, that's probably easy. I'm not so sure yet. That by simulating that for complex equation with this Monge-Ampère coupling, you can interpolate between pure diffusion and that equation that has been derived by Bertrand Maury with a different model. And that I'm not completely sure, but that's the hope. And then you can really see that okay, it's not just a computational model. There is actually linked with actual PDs in the literature. Someone is calling you. So I will conclude here. So okay, so we have a fast and simple way to generate soft body with some constraint on the shape and volume. There is this algorithm with the internal pressure that's actually called Lloyd algorithm, I forgot to say. It's already in itself almost an active matter model. You can just add all the forces you want, and then the game is to choose the forces. And most of the classical biophysics test cases that I know, they fit into this framework with some appropriate modeling choices. So there are some drawbacks. So I said that you have to find a compromise between physics and math and everything. So here the math and computation is pretty easy. But the reviewers complain that maybe it's not so much physically grounded or as much as physically grounded as low resolution method like this finite element method, but you can input some physics in each part of your mesh. There are other ongoing works on how to fit that with data and how to implement these energy minimization approaches. So instead of defining arbitrary forces, I can just define an energy that would penalize the surface tension, the interaction between that would model surface tension. And then if I take the gradient descent of that energy on the parameters that I have, so the position or volume, whatever, then I should obtain something. The problem is that it's difficult because the model is the solution of an optimal transport model. So we have to derive the solution of optimal transport model, and I'm not sure I know how to do that. And then what are the links with continuing PD models? And I will stop here. Thank you for your attention.
I had a question about the implementation of it, because correct me if I'm wrong, but at the heart of the method is you need to have an actual discretization of the whole space to compute the.
In theory, no, but yes.
And when you do it in practice, how precise does it need to be, and how do you influence the actual computation, and how expensive is it?
The complexity is of the total. Is a big O of n is the number of cells you have, and m the total grid cells. So not per dimension of m to the d if you want.
Yeah.
So it's still quite expensive, but you can do it in parallel. That's the trick. So on the GPU, it's pretty fast. Without GPU, it's a bit.
Do you think it would be possible to imagine much bigger system with like, let's say a compact cost or whatever, so as to make multi-scale optimization of it?
That would be a good idea, yes, because in the end, most of the time you spend to compute is to compute the void, basically.
Yeah.
And that you would like to say, okay, I don't care. So I guess it's not that that would be the same, because the multi-scale method would mean that instead of discretizing the Lebesgue measure with Dirac masses with the same size at the given position, you just at some point you find a mesh which is bigger. I haven't tried, because then you have to find at each time how to rediscretize your Lebesgue measure. But maybe it could be possible, yes.
Yes, so maybe a remark on that, because it's a question that we discussed a lot, especially with the colleagues who do the metal, the steel structure stuff. So basically, it's doable to go in this direction. The thing is that it's very doable if you're working with isotropic cells. Basically, if you start to consider, for instance, very elongated cells, then it's less clear in terms of locality and orientation. So you are less incentivized to do it. And maybe an important remark is that it's O of nm, not O of m squared. So even if you have millions of voxels, you think, oh my God, it's quadratic, it's never going to scale. But even if you have millions of voxels, you still have at most a few thousand cells in practice. So it's not that bad.
So next is the discretization of the space, no?
Yes, but basically the complexity of the algorithm, it's linear with respect to the discretization of the space, not polynomial.
Yeah.
So even though when you see it, you say, it's never going to scale, it's going to be a huge problem. In practice, it's not that big of a problem. So that's why we haven't done it yet. It's because the incentive is not that strong to go in this direction. You know, I played a lot with things like Tai Chi, you know, try to work with other solvers. And you're like, oh, okay, maybe. So, you know, basically for this range of problems, it's not that bad. So you can scale, you know, it's more interesting to have this modeling freedom and work as Antoine is doing with different cell interaction problems. So actually, we didn't show pictures about that, but you have the people at Pixar who simulate like bubbles and splashes. They use the same models. Actually, they call them power diagrams, but it's the exact same math, and there's a strong link in the literature. And basically, I would say the one guy who invests a lot of time into going beyond those schemes, it's Bruno Levy, who came 2 years ago, and that's because he's working in astrophysics in a setting which is extremely degenerate. So in a sense, his motivation in physics, it's kind of the worst case scenario for those schemes, and so that's why he cannot discretize the space with voxels like we do. He has to use very clever data structures, and that's why his talk was, I think, quite hard to follow.
Yes. So here we use the discretization of the space, but I think in this simulation it's real semi-discrete obtain transport, so the space is not discretized.
Just to get an idea, how long is it to just make the small animation?
So in 2D it's no.
Sure.
Yeah, in 2D it's real time. In 2D it's, I mean, all these. So this takes a bit more time, right? Because also you have to add particles, remove, blah blah. So in 3D you may have to wait a few hours at some point, but all the 2D simulation I show, so this is basically real time.
And when I played with the GPU on my computer, it was not real time because I was adding too many shaders to have nice pictures. But
Yes.
if you don't care about that,
Yeah, the next bottleneck is the plotting.
I have a question about when you started to explain how to update the position based on the cost. So you, as I understand, so you update the position and then you recompute the diagram, the diagram.
Yes.
But could you fix the diagram, the tessellation, and update the xi? Because
But that's, I mean, we do one after all, right? You compute the tessellation, you update the xi, you compute the new one. That's it.
sure. No, but my question is because when you introduce this idea about xis and volume constraints, so of course, if the xis are very packed in a small area and the volumes are big,
Yes, you will have something very strange.
It's a tessellation, but the x not
Yes,
it's a position of the cells.
yes.
So in this situation, could you retrieve the natural position of the xis by just do you see what I mean? Because
Yes.
it looks like the optimal position xi would be in the center of the cells, not.
That's what Lloyd algorithm does initially, I think. So what you can do to start with something a bit realistic is for the few steps, you just put exactly what you did. You just you compute a Lagarde tessellation. You see that it doesn't look very good. So you just put the xi at the center of the computed Lagarde tessellation.
Because that's the optimal position?
No, because it looks more realistic, as you said.
Okay.
But
There's no optimal solution. That's
but I in terms of what? I mean, that's
for a given tessellation, is there different XI?
you want to optimize the cost itself. That's what is.
No. I'm taking the same cost, but
Yeah.
I want to fix the tessellation and optimize the XI for the same tessellation. Is there, because there's no one-to-one correspondence, or there is a,
But if you keep the same cost, the minimum would be the absolute minimum of that total cost,
sure.
right?
And for a given,
And for that, if you want to compute the absolute minimum of that, you do what I told you, which is Lloyd algorithm. You compute the tessellation, at each step of the algorithm, you put the xi at the center of the Laguerre tessellation you compute, and at some point you reach a fixed point, which is the minimum of that total cost. Oh, I'm not sure I answer your question.
I'm just wondering, because for given XI and prescribed volumes, you have a given tessellation.
Yes.
But if you, for this tessellation and these volumes, you keep these, but you try to optimize the XIs.
Yeah,
Is it
so
possible?
they are the centroids of the.
So they
Which
become the
cell?
centroids.
Yeah.
Yes.
I mean, it's,
Mathematically, that's a solution. Yeah, okay.
Yes. And it's basically how the, you know, like those references, Galouet, Levy, Bourne, etc. You have those small differences between, okay, you know that your cell positions, they would like to go to the centroids, but currently they are here. So what should you do? Should they teleport? And that's exactly Lloyd's algorithm for K-means. Or should you have a soft string that brings you there? And so the people at Pixar, they would teleport. And the people at Inarias-Saclay, they would add a soft string, because then with this soft string, they are able to show convergence of their schemes towards the real solution, etc. So that's, and basically, when I told you that Bruno Levy was in a much harder setting, it's exactly because of that. Because what Bruno Levy is doing is that you have the astronomical filaments. That's so you see the galaxies. So that's your points, and you want to match it with the Lebesgue measure on the 3D space. And so it's very degenerate. So you have basically the points are really on those 1D structures. They are not in volume. So points will want to move very far away and will have some very elongated Voronoi set. And that's much less, let's say, well conditioned as an optimization problem. But we, Antoine, in this simulation, he's in the easy setting because you have already moved your cell centers to be well spread in the immune space. So as far as optimal transport problem goes, it's an easy one.
So in your simulations, you never had the case that the XI go out of the cells.
Essentially, no. Thank you. Except if you have maybe if you look very carefully, I don't know. You don't put that. So in that kind of setting, when alpha is very small, so you have a very concave core, so it's almost flat. So you can have very weird shapes. And in that kind of setting, if you look very carefully, sometimes the centroid is not inside, but you don't expect that to be realistic anyway.
And do you think it's just a matter of the actual time step size that you take, or is it?
No. I mean, it's somehow a drawback of having a unique solution. It's just the theorem gives you that tessellation, and even if the centroid is not inside, it's like a cell that's how it is.
The interaction between the false solver and the tessellation. If you make really small time step, do you still get these issues?
I think so. Yeah. You can find configurations if the centroids are very close. It's clear that they cannot really be in there like I said. There will be weird things. So just that expect that in practice, So I initially I thought, but maybe that's kind of a big problem or not, but in practice you very rarely observe that for anything that looks realistic.
I have a very less technical question about the interpretation of the outcome results of your simulations. It's very satisfying, I think. And it's very reminding me of the game of life. You know, game of life. So it's the attempt of creating biological processes based on very simple rules for computational simulations. And what is very funny with this game of life after years of literature is that you have patterns that emerge that look very much like a virus that will infect a cell and reproduce, etc., with cells expelling patterns on the regular, this kind of things. And so since you have this nice phase diagram with different structures, I remember this slide where the blue cells like the orange cells, and it's mimicking some kind of phagocytes.
Ah, yes, yes.
Yeah, so based on the type of patterns you have found, do you think there are some that are mimicking any kind of biological system of your knowledge?
Yes, I think so. I mean, I hope. I hope. So basically, all these systems that are actual biological systems. So in, I mean, you probably know that even better than me. In embryogenesis, the only thing you have at the very beginning is just different types of cells, and they organize, and you can see this engulfment or a phenomena or sorting. So these are patterns that are observed in nature. So the question, if your question is, have we
it's
precise
like
data
in your simulations and phase diagram?
Do
you think you have found
for all of them? Probably not.
They were interesting.
Probably not. All the patterns exist in nature. No. Probably not all of them. But no. Yes.
How do you implement cell division?
Cell division is for like this, for instance, this one.
Any situation, right?
But for this one, it's really easy. You just so you start with a position, and then you pick a random point, and then you cut into, a random axis. Sorry, and then you
But how do you
kill
set
that.
it to just?
Oh yeah, it's random. So here the axis is random. If you have a preferred axis, you can implement your preferred.
So you
axis.
don't make your cell grow before.
They split when they reach a size, because when they divide, then you have two cells with half the volume, because you want to conserve the volume, and then they grow. And when they reach the appropriate size, with some probability, they divide along a random axis.
Can you also include some carrying capacity or constraints on the way that
You mean if the total volume
you
is
cannot have? I mean, there are some resources that have to be shared.
I guess that would be, yes, because so the parameters that you have is the time they divide, the axis doesn't matter so much. So you can always make the probability to divide depending on the environment. And the environment is either your tessellation or something external. So I guess that wouldn't be too difficult.
Okay. So probably we're good. Thanks again.
Let's go. Sorry, let's go. Can you show us the slide with the with the bubbles trying to escape through the hole?
Yeah, the very last slide. This one?
And there are people working on actually people escaping a room during emergency.
Well,
so I guess you could estimate the alpha for human or real people.
Maybe, yes. I guess, but somehow it's a very finite size effect. So it depends a lot on the. Yeah, okay. If for a given room and a given number of people and a given volume of people, there is probably an alpha for for these people.
You know, 2 years ago, there was a IG Nobel for rheology of cats.
Ah, yes.
Yeah.
So that's a very small alpha.
Sorry.